students. I hope all of you are fine. Today we are going to study the muscle contraction, and what we will study in the muscle contraction is the sliding filament model. Sliding filament model. I hope that you have attended the other lectures and you have a clear view of the structure of the muscle fiber and the muscle fibril. We are actually studying the detail of the functioning of the skeletal muscle. The sliding filament model is actually going to tell us how the skeletal muscle contracts. This is an explanation of the contraction of the skeletal muscle. It is going to tell us the various kinds of changes that take place during this contraction of a muscle. When we will be studying this topic, you have to remember what we have already discussed in the previous lectures. First of all, you have to remember the T-tubule, because the T-tubule is something that is very important in this case. You have to remember the T-tubule, you have to remember the sarcoplasmic reticulum, right? Then after the sarcoplasmic reticulum, we have to remember that we have something which was called calcium. We studied about something which was called calcium in the TNC, in the troponin molecule. We studied that the one TNC, one troponin molecule was attaching to the calcium. Then we have studied about the actin molecule, which is present in the lighter band. And then we have studied about the myosin. And in the myosin, specifically, what have we studied about? The globular heads. Now, all these things are going to be applied in this concept. If you have any problem, go back to the lecture, view the lecture again, and then come back to this class, because we are going to apply everything in this one. And then we will be studying about the H zone. I already told you what the H zone is. We will be studying about the H zone. Then we will study about the sarcomere, what the sarcomere is. We will study about the Z lines, distance between the Z lines. So it is very important that you attend all the lectures, you watch all the lectures that I have given you previously. I have explained it in a very, very easy manner for you people. Do go back if you have any problem, watch the lectures again, very short lectures of 10 to 15 minutes, and then come back to this lecture to understand the concept very, very well. Now, the sliding filament theory of contraction states that during contraction, the thin filaments, the actin, they kind of slide over the dark filaments. They slide over the myosin. This is generally the concept of the sliding filament model. What is it? That the actin filaments, the thin filaments, they tend to slide over the thicker filament, the myosin filament, and they overlap to a very high degree. If you remember the structure that we have already studied, we see that there is a kind of incomplete overlapping and it gives us a darker A band. But in the case of contraction, there is almost complete overlapping. And during this complete overlapping, various zones of the A band will disappear during the contraction. What happens now? That this is a whole procedure that takes place stepwise. In a relaxed muscle, the thick and the thin filaments overlap only at the ends of the A band, not complete overlapping. We say that there is an incomplete overlapping. I will go back to the slide and I will show you. But during contracted state, the overlapping becomes almost complete. The overlapping of what? The overlapping of the A band of the I band on the A band. It kind of moves over and completely covers the A band. The I band almost completely covers the A band. But this happens only and only when there is a stimulation from the nervous system. Here is a stimulation coming. This is the first point. Number one, stimulation from the nervous system. This stimulation from the nervous system arrives, and this stimulation it is going to stimulate what? It is going to stimulate the nerve fibers. The muscle fibers. It is going to stimulate the muscle fibers. And it is after this stimulation that what happens, that the contraction is going to take place. Now, when we go to a stepwise sequence of all these things, what happens that, first of all, we get a stimulation. Now, let us come to this point. I'm going to give you these things point-wise. Write down these points. The first point is the stimulation. Number one is the stimulation, which is coming from the nervous system. The stimulation can be because of a stimulus which might be present in the body or somewhere else in the environment, right? The muscle contraction is initiated by a nerve impulse which arrives at the neuromuscular junction. It arrives at the neuromuscular junction. Very important word. That is why I am writing it down. It arrives at the neuromuscular junction. The neuromuscular junction, as we can see from the name, it is a point where the nervous system and the muscles, they kind of join with each other, and the neurons are going to sign up. They are going to shift the message to the muscular system. They are going to shift the message to the muscles. Now, the nerve impulse is carried through the sarcolemma. I have told you now that nerve impulse, that change that has come is going to pass on through the sarcolemma. I told you about the T-tubules. The T-tubules are actually regions of invaginations in the sarcolemma. This whole message is going to pass into the T-tubules. And from the T-tubules, it is going to enter the sarcoplasmic reticulum. What is the sarcoplasmic reticulum? The sarcoplasmic reticulum is the endoplasmic reticulum of the muscle cell. It is known as a sarcoplasmic reticulum. What is it now? First of all, stimulation. Then the stimulation comes to the neuromuscular junction. From the neuromuscular junction, it crosses the sarcolemma. It enters the invaginations, which are called the T-tubules. And from the T-tubules, it goes into the sarcoplasmic reticulum. Inside the sarcoplasmic reticulum, we have special type of gateways. And in this case, the gateways that are going to be active are the calcium gateways. These calcium gateways, they will open up when the impulse reaches them. And when they open up, they will start to fill in calcium into the cytosol. They will start to pour in calcium ions into the cytosol, which is going to change the concentration of calcium in the cytosol. It is going to pour in calcium in the cytosol. Cytosol, the cytoplasm of the nerve fiber, okay, which is known as the sarcoplasm. It is going to pour calcium into the sarcoplasm, that is into the cytosol. When the muscle is at rest, the tropomyosin is arranged in such a way that it completely covers the actin binding sites with which the myosin will hold. But when the calcium concentration increases inside the cytosol, what happens is that TNC on the troponin, it combines with calcium. And when the TNC combines with calcium, it a little bit, it displaces the tropomyosin from its position. And as it displaces the tropomyosin from position, then what happens? The actin binding sites, they are exposed. The binding sites on the actin molecule, they are exposed. And these binding sites then, they attach with the globular heads, which are the cross bridges. They attach with the globular heads, which are the cross bridges. And this is possible only when the actin binding sites, they are exposed. And how are the actin binding sites exposed? They are exposed by displacing the tropomyosin. How is the tropomyosin exposed? Displaced, it is displaced when the TNC on troponin attaches with the calcium, which is coming into the cytosol. And where is this calcium coming from? It is coming from the sarcoplasmic reticulum. Once this contraction has taken place, then what happens? ATP is needed to hydrolyze and to break down the cross bridges that are formed. Right? Let us go to the presentation now and let us see what we have in the presentation so that it can become easier for you people to understand. Siding filament model. This is the dark band, this is the light band, this is in a relaxed stage, this is the sarcomere. And during contracted stage, we see that they have come close together. They have come close together and the H zone has reduced in size. This is the H zone. It is going to reduce in size in the contracted state. The sliding filament model was proposed by H.E. Hogsley and A.F. Hogsley in 1954. When a muscle contracts, the actin and myosin filaments undergo shifting and slide past each other. The Z lines are brought together. They are brought closer together. Now these Z lines, they come close together. The I band shortens. The length does not shorten, but the appearance shortens because it is overlapping. And the H zone disappears. This zone that is present during the relaxed state is the H zone. During contracted state, we do not have the H zone. The H zone disappears. The cross bridge is attached to the actin filaments and they pull the actin towards the center of the sarcomere. How the bridges are controlled in a resting muscle. The binding sites to the myosin are covered by tropomyosin. On stimulation, calcium is released from the sarcoplasmic reticulum. It binds with the troponin molecules, which move slightly and displace the tropomyosin, exposing the binding sites on the actin. ATP is hydrolyzed for attachment as well as detachment of the bridges. Here, now, this is the actin and this is tropomyosin, which is hiding the binding site. This is TNT, TNI, TNC. Once it attaches with calcium, it displaces the tropomyosin and it exposes this binding site. This binding site then holds the globular heads. During this formation, ATP is utilized both during the cross bridge formation and during the breaking of the cross bridges. This is going to be utilized in that situation. This is a sliding filament model. Now this is the I band, this is the A band, this is the I band. This is the H Z line, this is the Z line. This is the heel zone. And this is the M line, median line bisecting the center. This is in a relaxed state. This is the myosin, this is the actin. During contracted state, the H zone has disappeared. But once it gets relaxed again, this muscle is going to come into its own original position again. Now, this was all for the sliding filament model. If there's any question, you can ask me questions anytime. At rest, the tropomyosin covers the myosin head side of the actin molecule. During contraction, calcium binds with the troponin and displaces it. Myosin head binding sites are exposed, and ATP is hydrolyzed for the formation or the breaking of the bridges, of the cross bridges. The muscle contraction is initiated now, the whole division of the topic, by a nerve impulse. The sarcolemma of the muscle fiber, it penetrates deep into the cell, forming a transverse tubule. It's called the T-tubule, collectively the T-system. The nerve impulse passes into the sarcoplasm through the T-tubule. The calcium gates in the sarcoplasmic reticulum open, releasing calcium into the cytosol, and this calcium binds with troponin of the thin filament. The binding sites are exposed, and the cross-bridge formation and contraction takes place. Now, where does the energy for muscle contraction come from? The energy from muscle contraction comes from ATP, which is produced during aerobic respiration. Creatine phosphate is used in high energy requirement, but you should just remember ATP. Sometimes anaerobic respiration is also used. The muscle shape changes during contraction. Now, this was all for the topic. One question that can arise is that what is the cause of stiffening of dead bodies? The cause of stiffening of dead bodies. After death, dead bodies become very stiff. And why is this so? It is because after death, the ATP is absent to break down the cross bridges. The cross bridges cannot be detached. So the body becomes very, very stiff. And this condition is known as rigor mortis. However, after death, after 2-3 days, if the body is left or it is not yet buried, it starts to soften. It doesn't soften because of ATP formation, it softens because of the digestion by the enzymes that are present inside the body already. They start to break down the body, they start to break down the protein. That is why dead bodies become soft. The stiffness is because of the absence of ATP, which is going to break down the cross bridges. Now, controlling actin and myosin interaction by calcium, the same thing that is repeating over and over again for you people to understand it in one way or the other way to make it easier for you people. Contraction is initiated by the nerve impulse at the neuromuscular junction. I have explained this whole topic, and this is the third time that we are repeating it in the presentation. Anyway, you should listen to the whole lecture and try to understand the topic because this is a very important topic. All the muscle fibers are supplied by a single neuron to form a motor unit, and all the muscle fibers, remember, they contract simultaneously. They contract at the same time. Now look here. This is a muscle fiber, this is the neuromuscular junction, where the neuron is forming a junction with the muscles. And this is our muscle fiber. This is the motor unit. It is stimulating all the muscles at the same time, right? This is all for the lecture today. In the next lecture, I will be explaining to you people the nerve impulse and transmission of nerve impulse. See you people next time. Take care of yourself.